we look at the overall anatomy of the ear. Of course, we have the auricle on the outside. This is just designed for capturing sound. And I want to recognize, I want to make sure we understand this. When we think about sound waves, oftentimes we think about waves, maybe a sine wave or something like that, right? This would be a sine wave where the wave would go up and down like that. That's not the kind of wave we're talking about when we're talking about sound waves. Sound waves are compression waves. Sound equals compression. Or if we think about compression, think about pressure. This is important as we understand impedance. Impedance is an important. Well, we'll talk about impedance in just a minute. But as we try and maybe depict what a sound wave would look like, it really looks like this, where here's a bit of air, and then I'm speaking to you, and so there's a force generated, and that pushes this, and then that pushes on the next bit of air, and that pushes on the next bit of air. And so we propagate a wave based on pressure, as essentially that force pushes on the molecules in the air, and the movement of those molecules is what this pressure wave is all about. Okay, with this in mind, let's actually look at the different parts of the ear we got. Of course, we got the outer ear that we've already talked about. It's involved in capturing sound, and so in comes this pressure wave. Then it's going to work its way down the canal, and then we have the tympanic membrane, or what we essentially call the eardrum. And the eardrum is the boundary between the outer ear and then this section right here that we call the middle ear. And then we also have the rest of it that is the inner ear. And the inner ear can actually be subdivided into two different parts, one associated with hearing, and the other, equally important, associated with balance. And we'll go into detail about how that works. Okay, let's move away from this picture and go down to more of a cartoon picture. And here we have expanded those different portions, and I want to focus in particular on the middle ear. So as mentioned above, we have the tympanic membrane. It can actually be tightened or loosened by a muscle that attaches to it called the tensor tympani. And this is important because this acts as a protective measure. So the tensor tympani is sensitive to very rapid changes in the amount of force put onto the tympanic membrane. So if I'm exposed to a loud sound, if you're sitting quietly, and then all of a sudden, bam, you hear this loud sound come through, the tensor tympani will actually pick up on that and reflexively contract. And as the tensor tympani contracts, it tightens the tympanic membrane so that that huge pressure wave doesn't do damage to the membrane. This is where the idea of the toilet plunger comes into play. And what you can see now is the tympanic membrane is akin to me grabbing onto that toilet plunger and moving it back and forth. We'll talk about the purpose of that in just a second. But the rest of this middle ear plays out as the plunger itself. So if we look at these different ossicles, these tiny bones inside the ear, we have the malleus, the incus, and then down here, the stapes, or the hammer, anvil, and stirrup. The purpose of these bones is impedance matching, okay? The whole purpose of the middle ear is impedance matching. What does that mean? Well, when we think about acoustic impedance, this is more or less the same as resistance, and this is just the ratio of pressure and flow. To understand the purpose of the middle ear, we have to understand the fact that out here in the outer ear, what do we have? We have air. This is where the sound was generated. This is what we're hearing. But over here in the inner ear, way on the other side, we have liquid. And we'll see this in just a minute as we look at the cochlea and how it works. The problem is this. water is 800 times more dense than air. And as such, the impedance of water is about 3,500 times higher. If a sound is generated in the water, it travels very quickly. I can hear it very well. But imagine going to a swimming pool and listening to people. You're maybe just floating in the pool and you're listening to conversation that people are having at the deck of the pool, and then you go underwater. You can't hear them anymore. You might hear garbled noises, but you can't hear them. And that's because as the sound travels through the air and hits the water, the impedance difference between those two is very significant, such that that pressure wave cannot penetrate into the water. That's a problem because for our own anatomy, the pressure wave was generated in the air, but we've got to figure out how to get it into the liquid. And so when we think about this idea of impedance matching, it's all about amplification. I essentially need to amplify the pressure nearly 20-fold in order to get it to register on the sensitive parts of the inner ear. And it does that through this series of levers. In reality, the stapes, even though it's not drawn accurately here, the stapes is much smaller than the tensor tympani. So this allows me to take all the pressure hitting the tympanic membrane. All the pressure hitting the tympanic membrane can be channeled and leveraged through this series of bones onto a very small stapes, which then acts as a toilet plunger. The stapes is directly connected here to the oval window, and as the tensor tympani moves back and forth, this whole range of bones makes the stapes also move back and forth, just like the plunger on a toilet. And this generates a pressure wave inside the inner ear that is amplified by this series of inner ear bones, the malleus, the incus, and the stapes. Okay, now that we have that movement, what actually happens? So we come back to this structure here. This is essentially the cochlea that, rather than being in a big, huge conch shell shape, we just unrolled. And this is what we see. We see the oval window here, the round window here. And please, again, appreciate, this is a sealed container. There's no actual hole in this process. The oval and round windows just represent membranes that are flexible. And it's important to have two because it's a sealed system. If the stapes is pressing inward on the oval membrane, that's generating pressure on the inside. That pressure has to be relieved somehow, and it's relieved through the round window. So as I push in on the oval window, the round window tissue will bulge out, and vice versa. And these two things will work together back and forth. Now we have here, this is the basilar membrane. The basilar membrane is thick on this end, but narrow, and on this end, thin and broad. Very much like, I don't know, if you're a musician, think of a double-reeded instrument like an oboe or a bassoon, how these reeds are cut to be very thin, and the broadest point of these reeds is at the end. The whole idea behind that is to pick up resonance vibration within that structure, and that's exactly what we see here. So how does it work? So we have the stapes. It's, of course, with the sound, it's going to plunge in, okay, and then it's going to move back out, and this is going to create a wave, a pressure wave inside this structure. We talked about, especially with a high frequency, high pitch sound. So this is rapid movement, and what you end up getting is a wave. The first thing that the thing's going to do is going to dive down, because that's where the exit, the round window, is. Then it's going to come back up, maybe right there. So we see right here, this is the resonance frequency of a high frequency sound, and therefore high pitch. Something with a low frequency would come in and find its resonance frequency on this end. This is going to be a low frequency and therefore low pitch. And then something in the middle, of course, would be medium frequency, medium pitch. That's the idea for how the cochlea works. There are places from the beginning all the way to the apex. each one of these represents a different resonance frequency. And as the stapes beats on the oval window, the pressure, the frequency of the pressure wave generated by that force will find its resonance on this basilar membrane and cause the basilar membrane to move just in that area. And as that area vibrates, that's where the neurons from the cochlear nerve will be stimulated, and they'll take that to the brain, and that will be identified as a specific pitch associated with sound, whether it's a B flat or a C sharp or 2 octaves higher than C, whatever it is. It's based on the location of the vibration, which is based on the resonance frequency associated with the thickness of the basilar membrane and its width. Okay, with that in mind, let's zoom out and let's see if we can figure out what's going on inside the cochlea to allow this movement. Okay, this picture down here is essentially if we were to take a cross section through this tube, this is the structure that we would get. So this top structure is the vestibular duct. This bottom structure is the tympanic duct. This green structure, very important, is the vestibular membrane. Here we have the tectorial membrane. The basilar membrane that we've been talking about is this area here that separates the two. So this structure here, basilar membrane. There are two really key things that we have to understand as we look at this structure. Number one, the fluids that fill these membranes are different, and this is going to be critical as we think about the overall job that we have of turning a physical stimulus, in this case a pressure wave, into an action potential. The vestibular duct is filled with perilymph. The tympanic duct is also filled with perilymph, but the cochlear duct, which is over here, this is filled, I'm going to put a star here because this is very important. This is filled with endolymph. And endolymph is different than perilymph in that endolymph is very, very, very enriched in potassium ion. And the cell structure that does this is this structure right here. This is called the stria vascularis inside the cochlear duct, and its job is to make endolymph. In other words, it takes perilymph and it exchanges sodium for potassium. That's essentially what it's doing. So there isn't a big charge difference here, but it's very, very, very enriched for potassium. And that's going to play a huge role in understanding how we get this action potential. The other thing that we really want to highlight here is this structure in blue. We've got the tectorial membrane already listed. This is part of it. But this entire structure right here is a structure that we call the Organ of Corti. And it's going to be, this is where the magic happens when it comes to sound. Just to give you a picture of that, we do have, for example, well, let's look at it this way. We have the cochlear nerve. The cochlear nerve is going to be coming, and the cochlear nerve is going to embed itself here. And this is where sensory cells, as part of the organ of Corti, here stimulate the cochlear nerve to get this action potential to the brain. This will join the vestibular nerve to become the vestibulocochlear nerve, cranial nerve 8. Understanding how the organ of Corti works is critical to understanding the function of ear or hearing. And we have to understand at a very minimum the difference between endolymph and perilymph, and I'll show you why that's so important right now. Okay, let's expand this organ of Corti. We'll draw it down here. This is essentially what we're looking at. Remember, we're sitting right here on the basilar membrane, and up here we have the tectorial membrane. Well, what we have as part of this reticular lamina, this area down here, this consists of hair cells. And we actually have one row of inner hair cells and 3 rows of outer hair cells. And the hair cells, this is very important. We have 1 row of inner hair cells, so this is an inner hair cell. And then we have typically 3, anyway, several rows of outer hair cells. This inner hair cell, this is what we're really focused on. If we look at how this hair cell is designed, it actually has, right up here on top, it's got a long branch that we call a kinocilium that's embedded into the tectorial membrane. If we zoom in on that cell, we get a cell that maybe looks like this, where we can see labeled once again. Let's zoom in on this. This is the kinocilium that's embedded into the tectorial membrane. And now I think we can start to appreciate how this is going to work. The kinocilium is attached to all of these other stereocilia on the tops. These are all attached through structures called tipplings. Tipplings just happen to be mechanically gated potassium channels. Now you can appreciate as the basilar membrane vibrates based on the resonant frequency, this whole cell is going to shake back and forth. And this kinocilium, because the kinocilium, remember, this kinocilium is embedded up here in the tectorial membrane. And that's going to cause shifting back and forth of this kinocilium. And because the kinocilium is attached to all of these other stereocilia through tipplings, that's going to lead to either an opening or a closing of these mechanically gated potassium channels. And so it's the vibration itself that opens and closes these mechanical channels. Now, a couple of things that are really important to recognize. Number one, if we go back and we look, the top of these cells here are embedded in this cochlear duct. And in the cochlear duct, we have endolymph. But the bottom of the cell is down on this other side in a tympanic duct where we have perilymph. So essentially, we can look at this cell and we can almost bisect it, where the top is endolymph and the bottom is perilymph. This is going to be critical to understanding now how this cell fires an action potential. We have, of course, down here our exocytic vesicles, all loaded with neurotransmitter, and we're waiting for the calcium signal to drive that, right? So as this kinocilium moves, it stretches open these tipplings. And because these are specific for potassium, this is the one time in the body where potassium actually is going to go in. So K+, K+. These ions are going to go in. Now, this is fascinating to think about. Why do we use potassium here? Well, because by transitioning to a single ion and its movement, we become much more quick and efficient in the processing of all of the information that's coming in. Of course, it takes a lot of energy to make the endolymph. The fact that the endolymph, remember, this is endolymph. And the key to understanding endolymph is that we have really, really high potassium concentrations. We haven't changed the potassium concentration inside the cell. Remember, before, whenever we would open up a door, potassium would leave because the outside was really low in potassium. But here it's different. Because the endolymph is so high in potassium, the concentration gradient is high to low, out to in. And it's the influx of these potassium ions, all of these positive charges, that leads to a depolarization event and the opening of voltage-gated calcium channels. So once these voltage-gated calcium channels are triggered, in comes calcium. Now, calcium is actually going to do a couple of different things. Like always, one of the things that it's going to do is cause exocytosis. So certainly calcium equals exocytosis of our neurotransmitter. This neurotransmitter is then going to be transferred Probably glutamate. I don't know that we know for sure exactly what this neurotransmitter is, but probably glutamate, and it's going to be transferred onto the neuron that I mentioned earlier that's part of the cochlear nerve. Now I've got my action potential going to the brain. But the second thing that calcium does is it triggers here the opening of a potassium, essentially a potassium leak channel. And when this opens, all the potassium that came into the cell from the top, now what does it do? It has a desire to leave. Why? Because the bottom of the cell is embedded in perilymph. There are tight junctions here that keep this cell isolated top to bottom. And because the bottom of the cell is in perilymph and the concentration gradient for potassium is now different because perilymph is so low in potassium concentrations, potassium wants to leave the cell. So in this scenario, we have one ion, potassium, that simultaneously depolarizes the cell from the top end and then repolarizes the cell from the bottom end. So in summary, if we were to look at how we transform an external stimulus, in this case a pressure wave, into sound. We come back up here. That sound comes down the ear canal and starts to vibrate the tympanic membrane. As that tympanic membrane oscillates back and forth, vibrates like a drum, it goes through the middle ear as a mechanism for impedance matching or amplification of the signal, converting all of that movement into the movement of the stapes. The stapes is directly connected to the oval window and starts moving the oval window back and forth. That back and forth movement generates a pressure wave inside the inner ear, the cochlear duct, specifically, I guess, the vestibular duct. And as that vestibular duct receives that pressure, that all of that pressure it looks for its resonance frequency along the basilar membrane, and the basilar membrane starts to move. As the basilar membrane moves, specifically in reference to the tectorial membrane as part of the organ of Corti, the hair cells that are embedded in this start to vibrate back and forth. And as we look at these hair cells, again, that's a shot of how the vibration of the basilar membrane moving up and down is going to wiggle these cells back and forth because of the kinocilium embedded in the tectorial membrane. That movement in and of itself is going to open and close tip links. Now, this is kind of important to recognize as I've got drawn here, right? One direction opens and one direction closes. At rest, the tip links are always a little bit open. So this isn't a binary system where it's on or off. At rest, if we're not hearing anything, these hair cells are sending a specific frequency of action potentials to the brain. And that frequency is essentially the background noise that communicates to the brain that the hair cell is functioning and working properly. In this way, I can help predict directional sound, because if you can appreciate a sound that comes in from your right, it hits your right ear sooner than it hits your left ear. And so the right ear is actually out of phase with the left ear in this opening and closing pattern. And so what the brain registers is a change in frequency. It's going open, close, open, close, open, close. On the other side, because I'm out of phase, this allows that instantaneous detection by the brain processing this information to determine the location of sound. And so through the corpora quadrigemina, specifically the inferior colliculi, this information is given to the inferior colliculi in the brain to cause that head movement to track toward the sound, and it knows where the sound is coming from because of how these things are, the right and the left ears are out of phase with each other in this process. Anyway, as the tip links open, potassium rushes into the cell. Potassium can go in because the top is embedded in perilymph, which is very high in potassium. Potassium depolarizes the cell, triggering voltage-gated calcium channels. Calcium comes in, and this time calcium has two jobs. Number one, it causes exocytosis of my neurotransmitter. And number two, it also opens up these potassium essentially leak channels that when they are open, because these channels are at the bottom of the cell, which is embedded or bathed in perilymph, potassium will leave, and that has the effect of repolarizing the cell, and then we get to start all over again.